Deep within the halls of the Sinai Medical Center, Dr. Aris stands before a glowing, high-resolution display. On the screen, a pulse of light flickers within bone marrow, revealing a hidden world. His team is hunting a ghost known as clonal hematopoiesis. It is a silent genetic mutation lurking in the blood of millions, waiting to strike. They have identified two specific culprits, the Jak2 and Tet2 mutations. These genetic errors are not just passive markers. They act like high-octane fuel for arterial plaque, silently accelerating the path toward heart failure and vascular collapse. The mission is clear: find a way to stop these rogue cells before the damage becomes irreversible. To solve this biological puzzle, the lab launched a rigorous 12-week study. They focused on mice carrying these dangerous mutations, dividing them into 3 distinct environments. The first group was given access to spinning wheels for voluntary exercise, allowing them to run freely. The second group faced a much harsher reality, exposed to sleep fragmentation through mechanical probes that disrupted their rest. The third group remained in a standard, controlled environment as a baseline. Day and night, the researchers monitored the cellular changes. They were looking for a breakthrough, a sign that lifestyle could override the hard-coded instructions of a mutated genome. The tension in the lab grew as the final data began to materialize on their monitors. When the results finally appeared, the contrast was staggering. In the sleep-deprived mice, the Jak2 mutated cells did not just grow, they surged. The screens showed a chaotic flood of inflammatory proteins invading the bloodstream. These rogue cells were triggering a violent chain reaction, forcing the body to attack itself. We watched as the arterial walls thickened, choked by the rapid buildup of plaque. It was a clear demonstration of how cellular stress, born from a lack of rest, can turn a manageable mutation into a lethal weapon. The biological brakes had failed, leaving the cardiovascular system vulnerable to an internal fire that seemed impossible to quench. Then, Dr. Aris pointed to the exercise group, and the room went silent. The results were nothing short of miraculous. The simple act of running on those wheels had stopped the clonal expansion almost entirely. The data revealed that exercise lowers bone marrow inflammation at its source. It effectively forces those mutated, aggressive cells back into a dormant, healthy state. It is as if the movement resets the system, one researcher noted. By analyzing the chemistry, the team mapped the mechanism. Exercise elevates norepinephrine levels. This surge of natural chemicals acts as a master programmer, resetting the inflammatory instructions of the immune cells. Simultaneously, we discovered that quality sleep prevents the activation of inflammatory receptors that lead to premature cell death. Sleep acts as the ultimate biological brake on cellular stress. While some mutations, like DMT3A, remained immune to these changes, the Jak2 and Tet2 variants were effectively contained by lifestyle alone. This was the proof they needed. The body possesses its own pharmacy, capable of suppressing rogue cells through the simple rhythm of rest and movement. The data from the UK Biobank confirmed this wasn't just true for mice. Human subjects who engaged in high-intensity activity showed significantly lower rates of these dangerous mutations. We are not just victims of our DNA. We are the architects of how that DNA is expressed. The power to fight heart disease was hidden in our daily choices all along. The research concludes with a definitive truth. Lifestyle is a genetic switch. By choosing to move and allowing ourselves to rest, we can actively silence the rogue cells that threaten our hearts. Your health is in your hands. Sleep well, move often, and take control of your genetic future.